So I'll speak on 4 topics. I'll start off with some words on environmental complexity, and then I'll go on to presenting some strategies, some nuanced strategies that have evolved in the microbial systems to deal with this complexity. And then I'll go on to describing how we can construct models that can predict responses of biological systems, and then explain why we can do that. And true to what has been said so far, I would subscribe to the idea that the next frontier is bioprospecting. And what that would mean is a lot of our basic and applied science questions have been already solved by microbial or just biological systems in general. And they figured out how to deal with high temperature, high pressure, growing in complete absence of oxygen to make methane. They've learned to respire sulfur and grow at sub-zero temperatures. So it's a whole range of capabilities that have evolved over 3.5 billion years of evolution. And there's no niche on Earth that we know to be completely sterile, so to speak. There might be some, but I don't know. Maybe John does. And the holy grail is to figure out the keystone organisms that live in these environments, so we can then begin to ask questions about how this complexity is encoded in the networks. We can figure out what these networks look like, and then we can rewire them into some form of a superbug that can allow us to then solve some of our pressing problems in bioremediation, bioenergy, and so forth. It's easier said than done, because if you go to one of these environments, such as a hypersaline system, one of the environments that I'm most interested in, you see that these are very large ecosystems. They have, they put together with desert and seawater, so they're plentiful on Earth, and they have huge potential in solving some of the huge problems we have in scaling up production of renewable, sustainable chemicals, say biofuels. But you can't grow E. coli and yeast in these environments. You really need to go and find the organisms that have learned to live here and then co-opt their strategies to engineer in your own interesting solutions. But when you look into these environments, or for that matter, any such environment, what you find is defining a keystone organism is non-trivial. And there are others who are more qualified to speak to this, so I won't belabor that point, but I will point out that in this environment, it's not just one organism or a few organisms, and it's not just who lives there, it's how they live together, and how they interact with each other that needs to be understood. And something Lee pointed out, a network of networks that can fully enable us to then re-engineer these systems. Now, I'll give you one example to just highlight the complexity of these interactions. So in this particular environment, the primary producer that assimilates carbon is eukaryotic algae called Dunaliella. And it takes the carbon in and channels a lot of it towards glycerol production, which gets stored in these vesicles. And you can see them as these green labeled particles in the cell. And the accumulation of glycerol can go up to 7 molar inside the cell, because these environments have in excess of 4 molar salt. So to balance the osmotic pressure, these organisms use glycerol. But when you stress them, and you can stress them in a number of ways, or if you put them in darkness, it was considered as a stress measure, although we see darkness every 24 hours every time. These organisms release the glycerol in large amounts. And if you look at the process, it appears to be some sort of degranulation and exocytosis that corresponds with production of caspase. And so we got very interested in this, and we started looking at it very carefully. And what was striking was that the process occurs at an enormous scale. So you have over 50% of the population dying off when night time hits, and then the entire population regenerates with 2 to 3 divisions. And it recovers. And it continues on and on and on. And it seems unlikely that any creature would kill itself for no reason. And you can't explain why it does this unless you begin to appreciate the environment in which it lives. And the other organism it lives with, among many others, is a halobacterium, which John referred to in his talk. And this organism is a photoheterotroph, which means it can use light to make energy, but it needs organic compounds to grow. It cannot assimilate carbon in the similar way that Dunaliella can. So it turns out whatever is being released by Dunaliella complements all the nutritional needs of these algae, of halobacterium. And for reasons unknown to us, when you co-culture them, the productivity of algae goes up by a factor of 2. So there seems to be a reciprocal benefit to killing themselves. Now, if you go to the laboratory and we take Halobacterium and we start looking at its molecular responses, and we try to understand why it is eliciting the responses, we have to appreciate not just the environmental factors that trigger it, but we also have to appreciate the other creatures that are responding to the same environmental regimes and then behaving in dynamical and complicated ways. So this network of influences on these cells has a lot to do with how we understand the responses. So we all know about strategies that have evolved to deal with the environmental change, transcriptional regulation, post-transcriptional regulation. There's a whole gamut of things. But it's the nuances in these strategies that makes each organism unique. And for that reason, we've heard the analogy to electronic system, which is a great one to appreciate that it's not just the genes, but how they're wired together. But in reality, biology is not electronics. So you don't have circuits that are hardwired and constant in time and space, something that Chris was referring to. The circuits are induced, the parts are made, synthesized, modified, reused, and there's all kinds of dynamical complexity that exists, which is not seen in electronic systems. So I will give you two examples to reinforce this point, that the whole idea of characterizing each part one at a time and then putting it together into a large circuit is not going to work. Or projecting understanding from E. coli and yeast and bacillus and some of the well-appreciated model systems onto the lesser studied organisms is also not going to work, because each organism is its own creature. So when an organism gets stressed, one of the first things it does is it tries to explore the space of solutions. And if it's not already encoded, it starts shuffling its genome. And this process has been well documented, and people have also appreciated the role of this process in the evolution of operons. So microbes tend to then streamline their genomes, get rid of things they don't need, and then get the genes that are functionally associated closer to each other, and then often they get transcribed as a single polycistronic transcript. And this is one example, succinate dehydrogenase. There are 4 subunits that make up this complex used in oxidative phosphorylation. You look across all sequenced aerobic organisms, what you find is that these genes are always together, and it makes sense because they form one machine. But each polypeptide in this operon is not identical to the other polypeptide, so the properties of these proteins are different, their turnover rates are different, so you would expect that when the environment changes, there has to be some fine-tuning that needs to happen to maintain the stoichiometry of the subunits. Now, the early clues we got when we looked at this operon throughout the genome are these binding sites. So these vertical bars that you see are binding locations of transcription factors that are peppered throughout the genome and often encoding sequences and operons. So one would argue, a skeptic would argue that your data is all bad. But an optimist would say that when there's something else is going on here, we should look at it more carefully. Right? So we then asked, can we get some more evidence that shows that these binding sites are functional? So we looked at expression profiles for the 4 genes across hundreds of environments, and for the most part, they're highly correlated with a few exceptions. So then we looked at the transcript structure, which is essentially high-density arrays looking at the overall signal strength across the operon. What you find is that transcription initiates here and goes through the operon and terminates. So all 3 pieces of data, the genomic arrangement, the co-expression, and the transcript structure, all attest to the operonic arrangement of these genes. But when you bring in dynamics to look at what happens to the overall transcript structure in high resolution, so you look at differential expression of these genes using the tiling arrays, which allow you to look at different segments of the same gene at very high resolution. You begin to see that when cells go from high oxygen to low oxygen, a promoter inside the coding sequence of succinate dehydrogenase B is induced. And it turns out in mitochondria, you see a difference in stoichiometry of this subunit when oxygen levels are shifted. And this is not an exception, and this is something we looked at only over the growth curve. So you can imagine all the other binding sites may have some role to play in some other environmental context, so we cannot rule out their functional importance. And then we looked at all the operons, so hundreds of operons in Halobacterium, and what we found, so what we did is we took the expression correlation as one score, and then we combined these 2 measurements and came up with the tiling score, which allows you to look at the segmentation of this operon at the transcript level, and then asked how many of these operons have such internally located transcriptional elements. And just over the growth curve, we see that 40% of all operons have conditionally located transcriptional elements. And I bet you this is a unique feature of this bug and maybe some close relatives, and something you might not reproduce in some other organism. So if I were to do an experiment, even in this organism, in Halobacterium, in high oxygen, in these environments, and try to use a model generated from that data to predict responses to anoxic conditions, which would be these environments, there's no way I could ever make a prediction of this element being triggered. So this nuance would never be captured. And we've gone on to look at other organisms, and this same feature seems to be a rule, not an exception. And the second mechanisms that organisms use to adapt is gene family expansions. And what you're looking at is a machine that transcribes all genes in archaea, and bacteria have a similar machine, although instead of these two general transcription factors, you have sigma factors. And what has been seen is that organisms that have adapted to dynamically changing environments, the sigma factors in bacteria, or the TBP and TFBs in archaea have expanded substantially. I saw a slide just swing by in John's talk where they've done archaeal genome sequencing for 60 or 70 archaea, and it seems to be a rule across all halophilic organisms, archaea, that have adapted to hypersaline systems because they have dynamically changing environmental context. So to understand the consequence of expansion, we applied a systems approach to measure all the interactions of the family members in Halobacterium. Excuse me. And we looked at expression consequences of deleting them, fitness consequences of deleting them, and looked at the protein interactions. And we put all of it together, and a remarkable story emerged. So after duplication, there are mutations that are selected both in the coding and non-coding sequences that transforms the network of these general transcription factors, these TFBs, and they control large subsets of distinct groups of genes which are overlapping. They also control each other by binding to each other's promoters, and they physically interact with each other at the protein level for forming heterodimers. It's so complicated that you can't ascribe any single TFB to a specific fitness profile in a given environment. So here are star plots. So each sector here, the height of the sector correlates to the fitness contribution of that TFB in that environment. And we looked at over a dozen environments in temperature, pH, and combinations of those and so forth. So the only way 7 TFBs can contribute such complex fitness landscapes can be if they were combinatorially. So we map genetic interactions between pairs of TFBs. And for one pair that I will show you, it was remarkable that the genetic interaction changed from a synthetic to a single non-monotonic when we went from 3 molar sodium chloride to 42 degrees Celsius. So the pathways in which these genes work changes depending on environmental context. Again, a point that I would say reinforces the idea that you can't look at one environmental condition and make predictions on other environmental conditions if you do one part at a time analysis. And what's more, if you change the regulation of duplicated TFB, so we took TFBD, we replaced its promoter with a related TFB, TFBE, and the expression differences are very minimal, and what we see is the fitness level just jumped up. So small changes to regulation of these expanded family proteins can have significant consequences on regulation and fitness. So the question is, how can we model such a complicated network when the environment has such a dominant role to play in genetic information processing at every step of what we know to be DNA to physiology or the central dogma. And there are 2 ways we can do this. One is, as I said, is you piece together each part by understanding each part one at a time. And I said there are issues with that, but you get a lot of valuable data from that approach. The second one, which is a systems approach, is to take the organism and perturb it in characterized ways and make measurements of all types of changes in gene expression, interactions, and so forth, and integrate all of this information to construct a model or reverse engineer a model that can explain how all the environmental perturbations were processed into phenotypes. And if you use mechanistic principles that are well understood, we consider dynamics and causality, then you can construct a predictive model. And there really are 2 major parts to this approach. One is to understand the modularity of the whole system, and second is to define how there's dynamic changes in modularity going from one environment to another using a regression type approach. So the first approach is essentially a biclustering approach that we developed several years ago. It integrates different types of information with the intent of finding genes or subsets of genes that are co-regulated over subsets of environmental conditions. And it does it iteratively by scanning the data space, by grouping genes and keeping them or deleting them depending on cross-validation score with some other orthogonal data types such as gene expression correlation. And this search space is so large that you have to guide the algorithm using other types of information, and we use two types. One is a network of interactions, so it could be physical interactions or functional associations from comparative genomics, and that allows the framework to then select the best set of genes to test in the next iteration. And since the goal is to find co-regulated genes, we also look for evidence of co-regulation, which would be shared transcription factor binding sites in the promoters. And there's other evidence also you can look for. So as the algorithm proceeds, it finds these signatures, and when it finds a significant signature, it will go and look in the genome for other genes that share that signature to check to see if they're also co-regulated with this regulon. So we see the environments being scanned and genes being scanned, and these motifs are discovered de novo, and the cluster eventually converges to about 30 genes in this example that are co-regulated over about 140 environmental conditions. And they have significant motifs that they share. And there's many ways you can use this. One, it groups genes of known function with genes of unknown function. So you can take a process that you well understood, and then you can expand it into genes that are poorly characterized, and there's many of those as you've heard before. You can also find different functions or genes from different functions grouping together in the same bicluster, and you can use that type of information to find regulatory linkages among different physiological processes. Okay. Now this is an NP-complete problem. What that means is the number of combinations in which you can group genes and environments simultaneously is so large that there's not enough computing time to find the same answer. And if you were to do it, you would exhaust all the computing resources and still get a less than optimal solution. So the way to get around this is by doing bootstrapping or an ensemble approach, where so the reason you need to do bootstrapping, I should point out, is that in every bicluster you might have wrongly grouped some genes that do not belong there just because of stochastic noise, and you might have not grouped genes that belong in the bicluster because you just missed it. And the consequence of wrongly grouping genes is significant on finding the signature. So the whole process is interconnected. So you can rerun this algorithm hundreds of thousands of times, which we do on the Amazon cloud, and then you have a space of associations amongst genes, the gene regulatory elements or motifs, and environment. So you can take a pair of genes and ask, how often do these two genes co-occur among the 100,000 biclusters that we've discovered? And when they do co-occur, which environments do they co-occur in? And when they are found together, what is the gene regulatory element that is discovered in front of these genes that is shared and might be responsible for their co-regulation? So you get a very large network which you can then process using community detection algorithms, such as finding friend circles in social networks or protein complexes in protein co-immunoprecipitation data. And then you can define this new organizational structure that we've discovered called Corems, or conditionally regulated modules. Operons are genes that are strung together in one polycistronic unit. Regulon is a set of genes that is regulated by the same transcription factor. Neither captures dynamics. Corems are essentially entities where genes are co-expressed, often because of the same regulatory mechanism, sometimes not, and their conditional regulation can be probabilistically defined both by gene membership and environment association and shared cis regulatory element as well. Here's an example of dimethyl sulfoxide reductase regulator, which is part of 3 different regulons, and the membership of this gene in these 3 regulons, or these corems, can be defined probabilistically based on the presence of motifs that are shared with genes in each of the 3 corems. So the DMSR is part of corem 1 because it shares GRE1, or regulatory element 1, with genes of that corem, and same with corem 2, it shares GRE2, and with corem 3, it shares a combination of the 2. And we can define the activity of these corems as a function of specific environmental conditions. So we can now say that, for example, the citrate lyase, which is part of 14 corems, when you look at 2 of the corems that it's a member of, it is part of 1 corem in these environmental conditions. They share expression profile in some conditions, but then it jumps to the other different corems. So the function of the genes is jumping from one functional group of genes to another depending on environmental context. And we can probabilistically assign a likelihood for how meaningful this is. And it turns out this captures a lot of functional information. And it probably will also explain why we often don't see the relationship between expression and fitness. We often think genes are upregulated or downregulated in a given condition. If it is up, it's important, and if it is down, it's not, and turns out that doesn't hold true. Instead, what we should look at is the Corem activity, or the mechanisms that are at play. So at 40 minutes after gamma radiation, we can now see which Corems are being turned on and off, instead of looking at which genes are turned up or down. Okay, and we can see specific functions that are emerging which can be associated with DNA repair and replication. Now we can say, okay, what's so unique about these Corems? And here I should point out that Corems are these nodes, which are genes, which are connected by different colored edges. So we can take this state that the cell adopts at 40 minutes post-irradiation and ask how it correlates with late phase of growth. And you see that there's completely new regulatory connections that are operating in late phase of growth, and the entire network reorganizes into a completely new state. So we can now do state-state comparisons for very specific spatiotemporal time points in the environmental responses of a cell. And we can ask what is shared and what is unique across these states. And remarkably, what we find is most of the Corems are shared. And there's few very specific Corems that are active that are associated specifically with an environmental response. And those associated with known functions, as I pointed out, DNA repair and replication for radiation response. So now the question is, these state transitions that you see in different environmental responses are being mediated by genetic and environmental factors. Can we capture those quantitative influences to build predictive models? And we can do this by regression, and we can say that the change in expression of genes in a Corem can be described mathematically as a combined set of influences from environmental and transcriptional factors and their changes in the same set of environmental conditions. And can give you quantitative models, and you can find examples of how, say in this case, oxygen changes, and in 5 minutes expression of genes in the Corem changes in a correlated manner. But there's a spatiotemporal aspect to it. And because it's a quantitative model, we can take as input just the measured values of these variables and then recreate the transcriptome profile of all genes in this Corem. And the predicted profile is in blue, and the measured profile is in red, and you can see that we can do very good, not just in the training data, but also in completely new environmental conditions. So this is a truly predictive model. And because these are, I'll skip on this, we can find the operational rules of metabolism because we can overlay these modules onto metabolic pathways. But this is not just for a few Corems that we can do this. We can do this for the entire network, which contains over 80% of all genes in the organism. And we have showed that this can be a predictive model for nearly all of the genes that are contained in an organism. And I should also point out that this model was constructed for an organism which was not E. coli or yeast, and in principle should be applicable to any organism that is now cultured from the environment. So the question is, why can we construct predictive models? And there really are some fundamental constraints that are imposed by evolution that make this happen. And I pointed out this in the first part as an issue of complexity, but actually this turns out to be very advantageous. So this is an example of an experiment we did with a diatom that we grew in the laboratory to simulate the coastal bloom. And all we did in this experiment was we let the cells grow and we put them in a day-night cycle. And as they grew, they utilized all the nutrients, and as they photosynthesized and respired in day and night, they changed the oxygen and pH levels. And so the entire framework of changes was highly coupled and complex. So although we might have changed one environmental parameter, all the parameters linked to this perturbation also changed in a physicochemically coupled and explainable way. And so for the cells, when they see such patterns, they can take advantage of it and evolve what is called anticipatory behavior. So for example, for halobacterium, if we put it through a day-night cycle and then we release it in constant darkness, we see that it begins to anticipate changes in oxygen. And you see it expects to grow oxically during nighttime and anoxically during daytime. And interestingly, it goes one step further and it anticipates oxidative stress as a function of anticipated changes in oxygen. And you see that the peroxyredoxin, which is an indicator of oxidative stress response, is induced at nighttime, and it continues to rise and through midday, where you have UV radiation-mediated oxidative stress. So you see responses that are 2 steps removed from the original perturbation. And we can think of this as a coupled architecture where the physiological responses associated with light, with oxygen and oxidative stress inside the cell are highly coupled in a way that they couple in the natural environment. And for this reason, you have limited degrees of freedom in which a cell can operate because all these gears are tightly coupled, so you can only turn them in so many different ways. And so they can't adopt infinite states, and rather they adopt a few dominant states, something which I showed you in comparison of late growth phase state and gamma irradiation state. And this is why, fundamentally, we can predictively model biological systems. Now, I'll skip through this slide, but I will point out that because we can do this using just gene expression data, if you can culture the organism in the laboratory, you can generate a model that is predictive and can be used subsequently for doing high-throughput synthetic biology. And going from complex microbiome, we can generate a model that covers over 80% of all genes in a newly discovered organism to drive expansion of the registry of standard biological parts, precise systems optimization, and also rationally directed evolution of microbes and their communities. With that, I'll stop, and in the interest of time, I won't go to specific acknowledgements, but the whole team was required to do this work, so thank you.